Hello everyone and welcome to the part 2 of our video lecture for Module 11, the Vibrio, Aeromonas, Plesiomonas, and Campylobacter species. So for part 2, our lecture will be all about the laboratory isolation and identification of these particular miscellaneous enteric pathogens. So again, here are the general characteristics of most of our miscellaneous enteric pathogens. So we will be dealing with the laboratory isolation and identification of this particular family or genera, or what we call the VCAP. So let's start with the genus Vibrio. So under specimen collection, for extraintestinal specimens such as body fluids, pus, tissues, or wound swab, we usually use the Kerry-Blair or autoclave seawater or the VR medium or the Venkatraman Ramakrishnan medium. So all of these can be a transport medium for increasing the viability of our Vibrio species, especially if there will be specimen delay, lalo na kung yung specimen collection was done outside the laboratory. So we can use this transport medium to increase the viability of such organisms. If, for example, we receive naman a stool specimen or fecal specimen, we can use blotting paper or strips of blotting paper and soak it in the liquid stool and place it in airtight bags with a few drops of saline. Usually, with this method, we can increase the viability of Vibrio species for up to 5 weeks. So just take note lang ha, for buffered glycerol silane, this is not recommended for the enrichment or for the transport of Vibrio because glycerol is toxic to most of the enteric pathogens, specifically your Vibrio. So for plating media under genus Vibrio, for sheep blood agar and chocolate agar, they appear as medium to large colonies that appear smooth, opaque, and iridescent with a greenish hue. So later on, we will show you that greenish hue. For sheep blood agar, we check for hemolysis kasi most of our organisms under Vibrio may cause hemolysis on the blood agar or can lyse RBC. For nutrient agar, there is not much clinical significance, but some organism may produce bluish tinge and transmitted light when grown on this particular culture medium. So later on, we will show you this particular Vibrio species. So for our culture media that is selective for Vibrio, we have your thiosulfate citrate bile salt sucrose agar, or what we call the TCBS agar. So this is the most commonly used culture media for Vibrio, specifically to differentiate the family into sucrose fermenting and non-sucrose fermenting. So for sucrose fermenting colonies, the color of their colonies is yellow, while the non-sucrose fermenting is green. So here is Vibrio cholerae on blood agar plate. So initially, it is surrounded by the greenish hue that is produced by the bacterium, but after a while, it becomes clear just like on this middle. So nagkakaroon ng clearing of the medium due to hemodigestion. Here are some Vibrio species on nutrient agar plate. So for Vibrio harbaei, this one is the species that produces parang bluish hue on nutrient agar. Okay? The rest, they appear as mucoid and large colonies. So here are the Vibrio species on TCBS agar. Again, for TCBS, we are dividing the genus into sucrose fermenters and non-sucrose fermenters. So for sucrose fermenters, such as your Vibrio cholerae, their colonies appear yellow in color. While for non-sucrose fermenters such as your Vibrio parahemolyticus, they appear as green in color. So we have your sucrose fermenter here, or the yellow colonies, and the green colonies or the non-sucrose fermenters. So in studying this particular slide, it is smart to just study the non-sucrose fermenters kasi mas konti sila. Kung titignan natin, we only have your Vibrio mimicus, Vibrio parahemolyticus, and Photobacterium damsela. For Vibrio vulnificus, it may ferment or may not ferment sucrose. So, very variable yung kanyang growth on TCBS agar. So here are the other plating media that can also be used in isolating and cultivating our Vibrio species. So next we have the maconchi agar. So on maconchi agar, they appear colorless, so they are non-lactose fermenters, but there are some organisms that may ferment lactose, such as your Vibrio vulnificus. And some sources says that Vibrio cholerae may also ferment lactose. So late lactose fermenter daw siya that appear reddish in maconchi agar. But this is quite questionable because this is from another book, okay? Sa ating Surinder Kumar book, on Mahon, this Vibrio cholerae was not mentioned to be a lactose fermenter. So for now, we will be considering this information as questionable. So wag muna nating i-consider si Vibrio cholerae as late fermentation, late lactose fermenter. So, but just want you to know that there are books that might be might consider this particular organism as a lactose fermenter. So, for your lactose fermenter, we only have one, the Vibrio vulnificus. Later on, I will show you. Next, we have the alkaline bile salt agar or BSA at pH 8.2. So, this is just the modified version of the nutrient agar medium. So, meron siyang additional 0.5% sodium taurocholate. So, this increases the viability and the recovery of our Vibrio species. The colonies is similar again on nutrient agar. So, they appear large mucoid colonies on BSA. Next, we have the Monsur's gelatin taurocholate trypticase tellurite agar or GTTA. This is commonly mentioned sa most of the books. So, aralin natin to because on this particular agar, the species of Vibrio may appear translucent with grayish-black center and a distinct appearance of a turbid halo. Ito. So, parang ganyan. So, black color yung colonies. Tapos, nagkakaroon ng turbid halo surrounding the particular medium. Next, we have your enrichment media for the Vibrio species that can be used for the preliminary enrichment purposes. So, first, we have your alkaline peptone water. This is useful for Vibrio species from stool or fecal specimen. So, next, we have the Monsur taurocholate tellurite peptone water at pH 9.2. This is also, again, an enrichment medium for Vibrio species. Wala siyang pinipiling species, okay? So, most of the time, this is general and not selective. Next, we have the chrome agar Vibrio. This is more of a selective one because it enhances the recovery of our Vibrio species, specifically your Vibrio cholerae, Parahemolyticus, and Vulnipicus. Next, we have the microscopic characteristics of your genus Vibrio. So, they are a sporogenous, so that means they do not produce spores. They are gram-negative, comma-shaped rods. Okay? Mukha silang comma na parang pag-ganon. Parang pag-ganon. Yan. Most of the time. Parang silang comma-shaped. They have polar, sheeted, monotrichous or multitrichous flagella. So, paano nga ba yung flagella nila? For monotrichous, parang ganito lang. Okay? Parang sperm cell. Ayan. Mono. So, single trichous. For multitrichous naman, polar pa din, pero madaming flagella. Okay. Ayan. Multitrichous. So, that means the particular organisms are all mostly motile. There are some organisms under your Vibrio that contains lateral flagella. So, pag sinabi nating lateral flagella, the flagella is on the sides ng bacterium. So, sa side siya. So, with this, if a particular organism has lateral flagella, they are mostly have the capability to swarm the particular culture medium, such as your nutrient agar. This is mostly seen on your Vibrio alginolyticus. Here are some biochemical characteristics for genus Vibrio. So, some species under this genus are halophilic or salt-loving, except for Vibrio cholerae and Vibrio mimicus. So with these two types of species, they can only tolerate at least 1 to 3% of salt or sodium chloride. So next, some of these species under this genus are catalase positive, oxidase positive. They can also reduce nitrate to nitrite except for Vibrio mechnikovi. All of them are susceptible to vibriostatic compound O129 and are positive in string test that is observed as mucoid stringing. So, ito yun. So, I believe nagawa na natin to. Kung hindi man, parang naalala ko, nag-string test tayo. So, the positive string test is the parang stringing or mucoid stringing appearance after mixing the colony to a particular solution. So, later on, we will show you what is that. This is the vibriostatic compound. So, as you can see here, nagkaroon ng zone of inhibition. So, that means yung particular organism is susceptible. At least, eto. Okay. So, pero kung titignan natin, maliit na lang, ne? Hindi siya super, malaking susceptibility result. So, this is slowly becoming resistant to that particular compound, which is the O129. So here is the string test for Vibrio species. So this is usually done on a glass microscope slide or sometimes plastic petri dish by suspending a colony, usually 18 to 24 hours old, to a 0.5% sodium deoxycholate. So the principle of the test, if a particular organism have cells that can lyse the sodium deoxycholate, then the suspension will lose the turbidity. So nagkakaroon ng viscosity yung solution, hence there is parang a stringing appearance of our solution or the mixture of our colony and sodium deoxycholate. So DNA will be released from the lysed cell, causing the mixture to become viscous or viscous. So the positive test for string test is, or positive organism rather, is Vibrio. Negative is Aeromonas and Plesiomonas. So this is a good test to differentiate Vibrio from other VCAP or other enteric pathogens under your VCAP. So the next test is oxidase test. This is used to determine if an organism possesses the enzyme cytochrome oxidase. So there are a lot of variation for this oxidase test, and the one that is flashed on the screen is what we call the filter paper test. So basically, we just submerge the filter paper onto a 1% solution of tetramethyl p-penylene diamine dihydrochloride. So after that, we just treat the particular colony of bacteria to that filter paper, and within 5 to 60 seconds, there is, if a particular organism has this cytochrome oxidase, the appearance of deep purple hue is observed. Negative is no color change more than 1 minute. Next, we have the gelatin liquefaction test. So this is used to determine the ability of an organism to produce extracellular proteolytic enzyme called your gelatinases. So if a particular organism have this enzyme, they have the capability to liquefy gelatin, which is present on our culture media. So the procedure for this particular test is to inoculate the gelatin dip, such as here on this picture, with 4 to 5 drops of 24-hour broth culture. Next is to incubate these tubes at 35 to 37 degrees in ambient air for up to 14 days. So for the next 14 days, we remove the gelatin tube daily from the incubator and place them at 4 degrees Celsius or ref temp to check for liquefaction. So the positive result for your liquefaction test or gelatin liquefaction test is the partial or total liquefaction of the inoculated tube, usually seen at 4 degrees Celsius. So for your positive organisms, we have Vibrio, S. aureus, Serratia, Proteus, and Bacillus. Negative is the complete solidification, just like here on this picture. So here are the Vibrio species under the carbohydrate fermentation test. So hindi lang cholerae. So Vibrio species, especially the 4 clinically significant ones, so almost all of them can ferment glucose, with Vibrio cholerae that can ferment sucrose, especially pati yung ating Vibrio algolinicus. So sila lang dalawa ang makaka-ferment ng sucrose or what we call the sucrose fermenters on your TCBS agar. For Vibrio parahemolyticus, may special siyang sugar na pina-ferment and that is the arabinose. So siya lang ang nakaka-ferment ng arabinose. For vulnificus, it is the only lactose fermenter among all the clinically significant Vibrio species. So we also need to take note of the cholera red reaction. This is the result of having the strongly indole positive result and reducing nitrates to nitrite, specifically for your Vibrio cholerae. So this is tested by adding a few drops of concentrated sulfuric acid to 24-hour peptone water, usually with culture at 37 degrees Celsius. A reddish pink color is developed due to the formation of your nitrose indole, such as here on this particular picture. So next we go to the decarboxylation of your amino acids such as lysine and ornithine by your Vibrio cholerae species. So again, what is the positive result for your decarboxylation of ornithine and lysine? So before that, where do we check first if decarboxylation yung ating test? We check for the butt area of our butt slant. So what is the color of our butt kung positive siya sa decarboxylation? So that is color purple. Well, for the aminase, where do we check? So we check for the slant area or slant part of the butt slant, medium. So anong positive result for the aminase? That is the red color of our slant. So that means for the aminase, yung slant natin kung positive color siya na red, that means there is acid formation all throughout the medium. While for the decarboxylation, so kung purple siya, there is alkalinity or alkaline, i-K na lang natin siya, all throughout the culture medium. So I hope that is clear. So the next are the tests for the differentiation of your Vibrio cholerae O1 biotypes. So again, who are these biotypes? So we have your classical and Eltor biotypes. So the first test is the chicken red cell agglutination test. So this is simply done by getting a slide and putting a loopful of bacteria on the middle of our slide. After that, we emulsify the colony by dropping a 2.5% erythrocyte, chicken erythrocyte suspension. So the indication of a clumping of erythrocyte is positive for Eltor while negative for classical biotype. Next is sensitivity to polymyxin B. So the organisms are tested by the disc diffusion method using discs containing 50 units of polymyxin B. So all strains of the classical biotype are sensitive while the strains of Eltor are resistant. So resistant yung Eltor natin sa polymyxin B. So that means, for example, sa isang MH8 agar, walang zone of inhibition yung ating anong tawag dito? Disc. So complete growth around our disc is observed. While for susceptible, like for example, dito yung ating disc, meron parang zone of inhibition. So that means the polymyxin B is strong against that particular organism. Next is the sensitivity to choleraphage 4. This is the most sensitive and most dependable test among all the three tests that we discussed. So pakipalitan to, no? For Eltor biotype, resistant siya, while the classical is susceptible. So these strains of classical cholera Vibrio are lysed by the routine test dilution method, while your Eltor are not lysed. So that means they are resistant to this particular bacteriophage. So here is the summary of tests and procedures that differentiate classical cholera and Eltor cholera biotype. So as you can see, for Eltor, positive siya sa lahat ng tests and resistant siya sa dalawang sensitivity testing natin. So, negative is resistant, no? So, palitan natin to. When we report sensitivity testing, mas maganda na we check for or we report them as resistant or susceptible. So, for classical, susceptible yung both polymyxin and group 4 bacteriophage or group 4 phage susceptibility test. So finally, we're on the other Vibrio species or what we call the halophilic Vibrios or salt-loving Vibrios. So first one is the Vibrio parahemolyticus. So it is capsulated, shows bipolar staining, and has a tendency to pleomorphism. So it may change shape depending on its strains. Next, we have your peritrichous flagella. So yung peritrichous flagella niya, just like this. So yung mga flagella are all over the particular bacterium. So, with this, it may have a tendency to swarm the particular culture media, such as here on this nutrient agar plate. So, for our culture media to use, we have your peptone water with 8% but not 10% sodium chloride. So, we can use this as an enrichment media for our Vibrio parahemolyticus. So, since salt-loving sila, so as much as 8% is required to increase the viability of this particular Vibrio. On ship blood agar, they appear as beta hemolysis. So later on, we will show you the picture of the beta hemolysis. Next, McConkey agar, they are non-lactose fermenters. So they appear colorless in the McConkey agar. On TCBS agar, they are non-sucrose fermenter as well. So their colonies is green in color. So here are the several biochemical tests to identify Vibrio parahemolyticus. So it is oxidase, catalase, indole, and citrate positive. It can also reduce nitrate to nitrite just like your Vibrio cholerae, and can also ferment a lot of carbohydrates, but the most important one is the sugar arabinose with the production of acid only. So it is the only Vibrio that can ferment arabinose sugar. It is also Voges-Proskauer negative, so pakipalitan to, negative siya sa Voges-Proskauer. It can decarboxylate lysine and ornithine, the amino acids, just like your Vibrio cholerae. So this is what I'm talking about, the beta hemolysis in Wagatsuma agar. So usually yung strains from humans are the one that causes hemolysis sa red blood cell. This is due to the heat stable hemolysin that is present or that present on our Vibrio parahemolyticus. Hence the name parahemolyticus, so they can hemolyze red blood cell. So what we call the Kanagawa phenomenon. So next, halophilic Vibrio, we have your Vibrio vulnificus. This is previously known as L-positive Vibrio or lactose-positive Vibrio or Benechia vulnifica. On TCBS medium, their colonies appear green in color, so that means they are non-sucrose fermenters. And just like your Vibrio parahemolyticus, they are Voges-Proskauer negative. The main characteristic of our Vibrio vulnificus is their ability to ferment lactose. So this is the only Vibrio that can ferment this type of sugar. They can also tolerate salt less than 8%. So nasa range sila ng 3 to 6%, but still they are halophilic or they require salt to grow on a particular agar. So for polymyxin B, they are resistant just like your L-Tor biotype of Vibrio cholerae. So next halophilic Vibrio is the Vibrio alginolyticus. So this is formerly regarded as the biotype 2 of Vibrio parahemolyticus. But due to phenotypic results or testing, it is now a separate species with a higher salt tolerance among all your Vibrio, more than 8%. It is also VP positive, which differentiate it from parahemolyticus and vulnificus, which are Voges-Proskauer negative. It can also ferment sucrose, which means that their colonies appear large and yellow colonies on TCBS agar. It is also more motile than your Vibrio parahemolyticus, so it can swarm the particular culture medium, especially the nutrient agar medium. So here are the tests to differentiate Vibrio parahemolyticus to its formerly subtype Vibrio alginolyticus. So we can differentiate them using Voges-Proskauer test where alginolyticus is positive, Also for sucrose fermenter, so alginolyticus is the one that can ferment sucrose. Also for swarming motility, and 10% sodium chloride, okay? So for your Vibrio parahemolyticus, negative siya because it can only tolerate until 8%. Ayan. For alginolyticus, it is high or strict halophile, so they can tolerate 10% sodium chloride. So here is another information differentiating your Vibrio cholerae, Vibrio parahemolyticus, and Vibrio vulnificus. So I got this online. So let's check if our information in the book is the same with this particular slide. So for oxidase, our Vibrios are almost positive. For the gas production, negative yung ating mga Vibrios. For lactose fermentation, for vulnificus, it can ferment lactose, but there are some sources that consider vulnificus as variable. So, it may or may not ferment lactose. Okay? For sucrose, we have your Vibrio cholerae, which appear yellow colonies on TCBS agar. Also for vulnificus, so it may or may not ferment sucrose. So, variable then. For inositol, they are all negative. For lysine decarboxylase, they are all positive, your cholerae, parahemolyticus, and vulnificus. Arginine naman, they are negative. While for ornithine, they are positive for Vibrio cholerae and parahemolyticus. Again, magulo si vulnificus, so variable yung kanyang result. For the growth of 0% sodium chloride or no salt content, only Vibrio cholerae can grow on this particular medium. So that means for Vibrio cholerae kasi, these are non-halophilic Vibrios. They can tolerate 1 to 3%, but most of the time, they can grow on no-salt culture medium. Unlike your Vibrio parahemolyticus and vulnificus, which are strictly halophiles, they cannot grow on lack of salt. So, dapat may salt content yung ating culture medium. On TCBS, their colonies appear yellow, just like we talked about dun sa sucrose fermenter. For Vibrio cholerae and for parahemolyticus and vulnificus, they both do not ferment sucrose. So this is from the book. Here is another table that can differentiate the groups of Vibrio species that occur in clinical specimen. So let's try to check their result. For no sodium chloride added or no salt content of culture medium, only Vibrio cholerae and mimicus can grow on this particular culture media. So they can grow without the help of salt. So they are non-halophilic Vibrios. For sodium chloride added, they can tolerate still, kahit na non-halophilic sila, for Vibrio cholerae and mimicus, they can tolerate at least 6% yung maximum. So more than 6%, they will not grow na kasi they are non-halophilic Vibrios. So for added salt, almost all your Vibrio species are positive. Oxidase production, so negative, meron tayong variable dito sa Vibrio mechnicovi. So this is uncommon, no? Hindi natin diniscuss to. It is not usually classified or not well studied, so negative siya. Pero almost all your Vibrio that are pathogenic in humans are positive. So for your cholerae, mimicus, and parahemolyticus, vulnificus are all positive for oxidase production. Reduction of nitrate to nitrite. So positive then, just like for oxidase production. Inositol, they are almost all negative, just like we discussed a while ago. While for arginine, check natin sino yung mga positive lang. Only Photobacterium damsela and fluvialis are positive in arginine dehydrolase. So again, hindi sila clinically significant, so hindi masyadong clinical importance yung arginine dehydrolase test. For lysine decarboxylase, so again, for your clinically significant, the cholerae, sino pa? For your alginolyticus, parahemolyticus, vulnificus, they are all positive for lysine decarboxylase, as well as your ornithine decarboxylase, positive for cholerae, positive then for alginolyticus or variable, sorry. So it may or may not become positive sa ornithine. For parahemolyticus, positive, and for vulnificus is variable. So for ornithine, only cholerae and parahemolyticus are can decarboxylase ornithine. So here is another differential test to differentiate the different species under your Vibrio genus. So Vaux-Prescauer, for clinically significant organisms, for Vibrio cholerae and alginolyticus, they are the only one that are Vaux-Prescauer positive. For Hemolyticus and Vulnificus, both are negative. While for motility, only Vibrio harvei is non-motile. For the acid production from sucrose fermentation, we have your Vibrio cholerae, which is a sucrose fermenter, and Vibrio alginolyticus, also a sucrose fermenting bacteria. For Vibrio parahemolyticus, so non-sucrose fermenter siya, again, green colonies. For vulnificus, variable, so it may or may not ferment sucrose. Next, we have your Celiobio, so not much clinically significant since yung ating Vibrio cholerae and alginolyticus are almost negative. So, hindi masyadong clinically significant. Except for vulnificus, which has a higher result for fermentation of Celiobios and as well as salicin. So, nice to know lang tong dalawang to. Baka lang lumabas sa exam. So, Vibrio vulnificus can ferment Celiobios and salicin. So, for presumptive identification, this is used to differentiate your genus Vibrio from your other enteric organisms or family. So, for susceptibility testing, your Vibrio are susceptible to vibriostatic agent O129. So, they cannot withstand this particular vibriostatic agent. And they're also positive to string tests, which differentiate them from the genus Aeromonas, which is resistant to vibriostatic O129 and are negative to string tests. Next is the inability to ferment inositol, except for Vibrio cincinnatiensis and some strains of Vibrio mechnicovi. So they cannot ferment inositol, and that differentiate them from Plesiomonas that can ferment this particular carbohydrate. Next, positive oxidase reaction. So this differentiate them from the negative oxidase reaction of Enterobacteriaceae. So may mga exemption, no? For almost exemption natin is Vibrio mechnicovi. So next is the fermentation of carbohydrates. So again, for Vibrio, they can almost ferment a lot of carbohydrates, and that differentiate them from the genus Pseudomonas that cannot. So here are the tests that can presumptively identify your genus Vibrio from your other miscellaneous enteric pathogen. So let's try to check these different types of tests. So for gram stain, hindi siya masyadong ginagamit because these three genus, all the same, are gram negative as well as oxidase positive. So dito tayo sa O129, susceptibility testing. So Aeromonas is resistant while the Vibrio and Plesiomonas are susceptible. For growth in TCBS agar, only Vibrio species can grow on this particular culture medium. For the growth of sodium or 0% sodium chloride, so hindi din siya ginagamit kasi variable yung Vibrio, while positive for Aeromonas and positive for Plesiomonas. 6.5% sodium chloride can be used because your Vibrio or some halophilic Vibrios can grow on this particular high salt content of culture media, while your Aeromonas and Plesiomonas are negative. For the acid production from the fermentation of such carbohydrates, so for glucose, hindi magagamit because they are all positive, as well as inositol. And for mannitol, only Plesiomonas cannot ferment mannitol. For sucrose, also Plesiomonas. And for gelatin liquefaction, only Plesiomonas. So, for definitive identification, pag sinabi nating definitive, so this is the reference or the standard, the gold standard for identifying your Vibrio species. So, kung gusto natin ng mas accurate na identification, we can use machines or technology and serological or molecular typing. So, the first one, we use the rapid and semi-automated identification system. So, we have three, the API-20E identification strip. So, hindi na natin to i-discuss, no? Kasi mababaliw tayo sa mga specifications nila. But all of these can identify more accurately the Vibrio species. The more sensitive, rapid, semi-automated identification system is the 16S ribosomal ribonucleic acid sequencing. So, this is the more sensitive among the three, while the matrix-assisted laser deabsorption ionization time of flight or MALDI-TOF mass spectrometry is the rapid and most cost-effective among the three. Ayan. So, pag nakita nyo yung mass spectrometry, mga ganyan yung mga RNA, so most of the time, these are identification systems. For molecular typing, we use pulse-field gel electrophoresis for surveillance or epidemiological studies, and some types of molecular procedures. For serological typing, we only have one, the use of polyvalent O1 antiserum, just like we discussed last time. So, this is used to differentiate the different subgroups of your Vibrio cholerae O1. So, we have your O1, the O139, and the non-O1 subgroup. Ayan, so finally, we are done with the lengthy discussion on your Vibrio species. Now, let's move over to another genus, which is your Campylobacter and Helicobacter species. So, Helicobacter was not mentioned sa ating title because this is a Campylobacter-like species. So, kamukha niya yung Campylobacter in terms of general characteristics and as well as biochemical tests. So, but we need to differentiate these two types of genera. So, both of them are asaccharyllytic, so they do not ferment carbohydrates. They are also oxidase positive, which is the same with your Vibrio species. And yung hallmark nila or general characteristic nila is that they are microaerophilic. So, they require less oxygen in their cultivation. Specifically for Campylobacter, requires 5% oxygen, 10% carbon dioxide, and 85% nitrogen. So, yan, 5, 10, 85. While for your Helicobacter, they require 5 to 10% oxygen and 5 to 12% carbon dioxide. Another thing to remember on this particular genus is that their optimum temperature to grow is 42 degrees Celsius. So, slightly higher than of a normal temperature for your bacteria. So, ang normal natin kasi is usually 35 to 37 degree or the normal temp for your incubator. But for your Campylobacter and Helicobacter, they require 42 degrees Celsius. That is there's an exemption for this. We have your Campylobacter fetus, subspecies fetus, which requires only 37 degrees. For flagella, they are all motile. So, for Campylobacter, it has usually single polar flagella. So, parang sperm cell. So, one flagella lang. While for your Helicobacter, it may have single or multiple polar flagella. So, madaming flagella sa iisang pole. So, now, let's go to the specimen collection and transport. So, this is just the same for your Vibrio. For Campylobacter and Helicobacter, we can also accept blood culture media or specimen that came from blood culture bottles. We can also accept stool samples and rectal swab but are not preferred due to the low yield of growth for this particular genus. For stool specimens, specifically if there is a delay in transport, we can use the classic Carey Blair for this particular specimen. So for gastric biopsy naman, specifically for patients with potential H. pylori infection, we can use the Stewart medium instead of the Carey Blair to increase the viability of H. pylori. For tissue samples, this is specifically only for this type of sample. We can use the cystine brucella broth with 20% glycerol. Normally, glycerol is toxic to enteric pathogen, but if the particular sample came from tissue, such as gastric biopsy, we can use the cystine brucella broth with 20% glycerol and can be stored at frozen temperature as much as negative 70 degrees. Again, glycerol is not recommended, especially for stool sample, because this is toxic to the Vibrio, to the enteric pathogen. So, for culture media, we have your Campylobacter blood agar or Campi blood agar. So, this was named in association with the genus that it permits to grow, your Campylobacter. If Campylobacter blood agar is not available, we can use Butzler medium and Skyros medium as well. If stool sample was received, we can use the Campi CVA, which has antibiotics of cefoperazone, vancomycin, and amphotericin B, which is toxic or can kill non-pathogenic enteric bacteria and only yield Campylobacters in the process. For selective media, we can use the charcoal cefoperazone deoxycholate agar or CCDA with the combination of charcoal-based selective media to increase the viability of your Campylobacter species. So if you want to increase the yield of the growth, much better na ito yung gamitin natin instead of the general culture media. So pwede natin itong gamitin to increase the Campylobacter species growth. While for H. pylori, we can use the chocolate agar or Brucella agar with 5% horse red blood cell together with your Skyros medium. So this is from the book. Here are the selective media for the cultivation of your Campylobacter species. Again, we have your Campi blood agar, the Skyro, the Butzler, and the more recommended agar to increase the yield of your Campylobacter species, which is the CCDA. So for microscopic characteristics of your Campylobacter, they appear as curved, non-performing, gram-negative rods. Also, they appear as long spiral or seagull wing shape in morphology. So later on, we will show you the seagull wing characteristic of this particular genus. Next, on gram stain, they stain poorly. That is why carbolfoxrin instead of safranin is recommended to be used as a counter stain. So just a quick review again for your gram stain. We have your crystal violet, iodine, alcohol, and safranin, or what we call the CVAS. Diba? Naalala pa ba? So crystal violet is used as a primary stain. Iodine is used as a mordant. Alcohol is used as a decolorizer. And safranin is used as a counter stain. So instead of the safranin, we use carbolfoxin as counter stain to increase the staining characteristics of our Campylobacter species. On hanging drop and phase contrast microscopy, they have the motility of darting characteristic. So parang ganito sa picture or video. So if you know the game darts, so it's as if you are throwing darts. So ganun yung motility nila. So parang iniumpog nila yung sarili nila sa microscope. Ayan. So ang cute, 'di ba? If hanging drop or phase contrast microscopy is not available, we can observe the motility or growth using Brucella or tryptic soy broth. So here is the classical seagull wing shape of your Campylobacter species under your gram stain. So saan yung mga seagull wing shape dyan? Ito mukha silang seagull wing. So ayun, nasa ano na yan, imagination mo kung mukhang seagull nga. Pero for me, mukha namang seagull wing tong ano na to. Rod na to or gram negative rods na to. So here is the summary of the biochemical test to differentiate your Campylobacter to your Campylobacter-like species such as your Aeromonas and Helicobacter species. So pinaka-importante dito is your Campylobacter jejuni, Campylobacter fetus subspecies fetus, ano pa? Helicobacter pylori. And as well as, syempre, add na natin si Helicobacter butzleri para meron tayong differentiation. So, pinaka-importante itong apat na to, but of course, on your board exam, it is much better to memorize the different characteristics of some of these species of our Campylobacter, Helicobacter, and Helicobacter species. So, try nating tignan kung saan sila nagdi-differ. For Campylobacter jejuni and Campylobacter fetus, they differ on indoxyl acetate hydrolysis. So, negative C is fetus, positive C Campylobacter jejuni. So, help niyo ako ha kung saan pa sila nag-iiba. Hipurate hydrolysis also, positive C jejuni, while for fetus is negative. Growth at temperature, positive si fetus at 25. So, mas optimal yung ano niya. Yun nga yung sinabi natin, di ba, kanina. Normally, they grow, for your Campylobacters, they grow at 42 degrees Celsius. So, kung yung Campylobacter jejuni natin, ayan, 42 degrees Celsius, while your fetus, this is the only exemption under your Campylobacter, which usually grows at only 25 degrees Celsius. Next, for ano pa? Nalidixic acid susceptibility test. So, positive si jejuni, negative si fetus. So, madami silang kaibahan. So, kahit yung growth lang sa temperature, ma-differentiate natin yung dalawang species under your Campylobacters. For Helicobacter pylori, ang pinaka hallmark is the positive urease test. So, among all the species under your Campylobacter and Campylobacter-like species, only Helicobacter species is the positive in urease test. So now let's head over to another miscellaneous enteric pathogen, and that would be the genus Aeromonas. So Aeromonas are gram-negative straight rods, oxidase positive, and are glucose fermenter just like your Vibrio species. They are classified into two groups based on their optimum temperature of growth, such as your mesophilic group, which usually has your species Aeromonas hydrophilia complex, Aeromonas veroni complex, and your Aeromonas caviae complex. So normally for mesophilic groups, they require at least room temp for their optimum growth, but they can grow until 37 degrees Celsius or incubator temperature. For psychrophilic group, they require colder temperature. So we have your species Aeromonas salmonicida, which is a fish pathogen, and most of our species here under your psychrophilic group are non-motile and grow best at 22 to 25 degrees Celsius. But since they are psychrophilic, so that means they can also grow at lower temperature as much as or as low as 14 degrees. So for culture media for Aeromonas, we can cultivate them on sheep blood agar, and most of our Aeromonas may exhibit beta hemolysis on this particular culture medium, especially species such as your Aeromonas hydrophilia, Aeromonas veroni biovar sobria, and Aeromonas jandiae, and as well as some strains of Aeromonas caviae. On McConkey, they are lactose fermenters, so this differs them from your Vibrio, which is non-lactose fermenter. So, a good example of a lactose fermenter Aeromonas is your Aeromonas caviae, which exhibit pink colonies in McConkey agar. So, to increase their yield or recovery for Aeromonas species, we can use a combination of your ampicillin with sheep blood agar with ampicillin and modified cephsulodin ergacin novobiocin or CIN plate 2. Usually, this CIN plate 2, the modified one, contains 4 microgram of cephsulodin, which is an antibiotic against a Aeromonas-like species, which is your Pseudomonas. So, in order for us to increase the growth of this particular genus, we can use this combination of culture media. There are some cases that the ampicillin pala, can inhibit the growth of Aeromonas caviae and trota. So, we can use SBA without ampicillin and in conjunction with our modified cephsulodin ergacin novobiocin CIN plate 2. So, again, for our cultivation of Aeromonas, we can use sheep blood agar without ampicillin and modified cephsulodin ergacin novobiocin CIN2 plate. In CIN1 media, so this is just the classical form of our modified CIN2 plate. So CIN1 media, there are your Aeromonas appear pink-centered colonies from the fermentation of mannitol with an even, clear apron-like pattern. So later on, we'll show you yung itsura nila sa CIN media. So here is the Aeromonas caviae in CIN media or the self-sulodine irrigacin novobiocin media. So they appear pink-centered colonies. So hindi masyadong clear dito pero parang nasa reddish pink siya. Pero pink-centered colonies due to the fermentation of mannitol. So they can also ferment this sugar with an even clear apron-like pattern or mas commonly from other sources, bullseye appearance. So, another example of your Aeromonas caviae exhibiting lactose fermentation on McConkey agar. Again, they are lactose fermenters, so they exhibit pink colonies on your McConkey. Here is a Aeromonas hydrophilia exhibiting beta hemolysis on your sheep blood agar. Here is the summary for the differential characteristics of your mesophilic Aeromonas species. So for this particular laboratory, we will be just focusing on the mesophilic group because these are the one that causes human infection. For your psychrophilic group, most of them are actually marine pathogens. So they are pathogens of the fish and other marine animals. So focus tayo sa mesophilic, especially for your Aeromonas caviae and Aeromonas hydrophilia. But of course, since konti lang sila, it is important for us to at least know the other species as well and their result on different types of biochemical tests. So now we're down with the last genus under our miscellaneous enteric pathogen, and this is your genus Plesiomonas. So it is now under the family Enterobacteriaceae, and the only species that is considered under this genus is your Plesiomonas shigoloides. This is a gram-negative bacilli just like your several miscellaneous enteric pathogen and are usually monotrichous or lopotrichous flagella. So, monotrichous, one flagella, lopotrichous, polar with multiple flagella. So, this is just like a polar multiple flagella. So, another term for lopotrichous. This is oxidase positive just like your Vibrio and DNA is positive. So, ito yung pinakakaibahan niya or distinct niya. And as well as TSI positive. So, in gram stain, for Plesiomonas shigoloides, there is not much clinical significance. So, parang the same gram-negative bacilli lang siya. So, it may occur in singly, pairs, or in short chains or filamentous forms. So, walang clinical significance or parang hallmark in order for us to identify that this is a particular Plesiomonas shigoloides species. So, for culture media or cultivation of your Plesiomonas shigoloides species, there is not much detailed information that was given on the book, so or sa kahit anong sources on our textbooks. But on online, for Plesiomonas shigoloides on blood agar, they appear as beta hemolytic colonies with distinct pink in color. So, parang ito. Ayan. So, we can also use the medium inositol brilliant green bile agar. So, they appear as pink colonies as well on this particular culture medium. On McConkey, they are non-lactose fermenters, so colorless. So that ends our laboratory discussion for our miscellaneous enteric pathogens. So here are the reference used for this particular video lecture. Thank you and have a nice day.